you very much for that introduction. So this afternoon in my presentation, I'm going to talk about the clinical pharmacology studies that are involved in drug development and how they help us to select the best doses for patients. Now, your first interaction with the clinical pharmacology group at the FDA is likely to result in us sending you this fairly lengthy list of items for you to consider during the development of your drug product. Now, at the outset, this can seem very shocking, perhaps even intimidating, to receive such a long, complicated list of issues, especially if you're new to the oncology drug development arena. But I can assure you that it's really not as bad as it seems on the surface of it, and the FDA is here to help you sort through all of this. Now, the earliest interactions that the clinical pharmacology group has usually begins just before you start your phase 1 trials in patients or volunteers. And usually the information comes to us in the form of a pre-IND or perhaps the IND, which would include the first-in-human study protocol. Additionally, you'll have the information you've accumulated around your product and a number of questions to the FDA regarding your plans and the suitability of those plans in the phase 1 trial. Now, you will certainly receive responses from us to your questions, and we'll probably expound upon other issues relevant to those. But we're also going to take the opportunity to send you this list of standard comments. Okay. So if you take a look at this particular list, this contains a number of items that we know can impact the clinical pharmacology behavior of your drug, depending on the type of product that it is and the route of its administration. And so you need to take a look at this and see how they may apply to the development of your particular product. Okay, so we'll also send you a list of guidance documents. Each of these guidance documents goes into more detail about the various items that have been raised on the comment sheet, and you can click on these hyperlinks and they'll take you to these documents so that you can have access to them, so you can better understand if they apply to your particular product and how. Now, the reason that we send these comments out early in development is to raise awareness about the issues that may be needed to be addressed during the development of your product. And we have found over time that this has led to better overall development of these products in the oncology area. So a very reasonable question to ask is what we're trying to achieve with all this. And really there are two things. So the first is the selection of the optimal dose for the patient population. And really we need to do this early in development. And I will spend a little time talking about why this is in a few moments. But the second issue that we are trying to address are dosing for patients who have some specific considerations. So for instance, some patients may experience drug-drug interactions and other patients may have hepatic impairment. And both of these kinds of situations can have an impact on the exposure of the drug in the patient, and that can have an impact either on efficacy or on toxicity. And so we need to try to understand these situations for these patients and to provide appropriate dosing to accommodate these changes. Now, when it comes to selecting the dose, we have an obligation to pick the safe and effective dose. However, we are also really aiming to find the optimal dose of the drug. And if we look at that in terms of the exposure response for any given drug, okay, we can have the relationship for efficacy plotted on the curve, shown in blue in this case, and the relationship for toxicity also shown in this case here in red. And we're looking for the exposure of the drug, and the dose of the drug, that really leads to the maximal efficacy for the drug relative to the amount of toxicity that is acceptable in that patient population. And this is how we define the optimal dose of the drug. Now, another question that you might ask is why we need to do this early in drug development. And the reason is really based on our experience. So we have quite a number of products that have been developed over time. And in many cases, we found that doses have been taken forward that turned out to be efficacious, but they also had a considerable amount of toxicity with them that impacted tolerability of the drug. And so we were left with the question of whether there was a lower dose that was still efficacious, that would have had less toxicity associated with it. And efforts to try and answer that question have been difficult over time. And so I want to use an example of BlendRep to illustrate two things, two good things that could be done during drug development, and how we end up with the situation I just described. So in the dose escalation part of development, they did really quite a wide range of doses, 0.3 to 4.6 milligrams per kilogram, which is very good. The other important thing they did is they selected 2 doses, which they took into their dose expansion trials, their efficacy trials. And this is very rare in oncology, and it's something that we really need to think about doing more of. Okay. Now, what they found was that, they tested 2.5 milligrams per kilogram and 3.4 milligrams per kilogram, and found they had comparable efficacy. But the 2.5 milligram dose had less toxicity, so that's the dose that was taken forward and approved. So these are 2 highlights of this program that I think are important, and we should think about doing more of in the future. However, like a lot of other drugs, we still found at the end of the trials that it had some toxicity that was higher than we would like to see. And we're left wondering whether a lower dose might have been better. In fact, in the dose escalation part, they did test 1.9 milligrams per kilogram. It seemed to have comparable efficacy, but there were only 4 patients, so there was really too few patients to make a determination about whether that's a better dose. So like many other drugs, we've been left in the position where we're asking the pharmaceutical firm to conduct more studies to assess the safety and efficacy of lower doses in the post-approval setting. Now, you might argue that paradigm is okay. However, another consideration is that trying to find the right dose in the post-approval setting can often be a very lengthy process. Okay, so here are a few examples of that. So Mylotarg was approved in the year 2000 at a dose of 9 milligrams per meter squared, and it was dosed on day 1 and day 15. But it turned out over time that this was a fairly toxic dose, and the efficacy was questionable. And at a later point, the drug company removed the product from the market and did some redevelopment of it. When they came back to us in 2017, they were able to demonstrate from the studies they'd done that 3 milligrams per kilogram on days 1, 4, and 7 was efficacious and less toxic. And this was the dose that is currently approved. So we finally got to the point where we found the right dose, but you can see, it took 17 years for us to get there. Another example is Marquibo. So this was a drug that the company tried to get approval for in 2005 at a dose of 2 milligrams per kilogram, but it didn't have the efficacy necessary to pass the trial. Another company then bought the product and further developed it and came back to the agency in 2012 with tests of the dose of 2.25 milligrams per kilogram and found that to be efficacious, and that was the dose that ended up getting approved. But that took an additional 7 years to get to that answer. And then the last example I have here is Jevtana, approved for prostate cancer at a dose of 25 milligrams per meter squared, which was efficacious, but the toxicity was quite high. And so we asked the company to go back and test other doses that might be as efficacious, but with less toxicity. And 7 years later, they came back to the agency with a study that showed that 20 milligrams per meter squared was efficacious and less toxic. And this is the dose that has moved forward. So here again, this took 7 years. So you can see that trying to find the answers to these questions in the post-approval setting can be difficult and very, very long. And so it really seems as though the onus is on trying to determine the best doses earlier in development rather than later. And this really begins in the first-in-human study, right, where we do our dose escalation. And the purpose of this is to determine what the recommended phase 2 dose is, the dose we want to take further into development to look at safety and efficacy. We should probably be, at this point, be considering taking 2 doses forward whenever that is possible. And the reason for that is as we get further into development, we find 1 dose is toxic, we will have tested a 2nd dose and know more about the safety and efficacy of that dose, which we may then be able to take forward. Another consideration is whether we should be targeting the maximum tolerated dose. And I think the answer to that is probably not. The MTD concept is really one we pursued when we were trying to develop chemotherapies some time ago. But nowadays, given that we're looking at targeted therapies like TKIs and monoclonal antibodies that react with specific antigens, we really ought to be thinking more about the optimal biological dose. That is, the dose that gives us the most efficacy with the least amount of toxicity. So when we see these kinds of applications at the agency, we provide a certain amount of advice. And so the first of those is really to make sure that you incorporate an adequate number of dose levels so that you can really distinguish between the activity and the safety of the product. If you get to the point where you have dose levels that are too close together and you cannot distinguish between those dose levels, you may actually need to pause and go back and add additional dose levels so that you can better see the difference between them. Furthermore, another important point here is making sure that you incorporate an adequate number of patients so you can make those determinations. Another thing to think about is the non-clinical data that gets generated. So in the past, when we were shooting for the MTD, we didn't really make the most of this information, in my opinion. But now that we're looking at targeted therapies, I think it makes a lot more sense to look more closely at the pharmacology data that's generated and the information that we might get on target engagement or receptor occupancy and that sort of thing, because they tell us about the mechanism of the drug. And so, for instance, if you had a monoclonal antibody drug and your plasma concentrations were perhaps 1,000, maybe 5,000 times higher than the saturation concentrations for the receptor, it might prompt you to think or rethink about whether you have the appropriate dose. So I think this information can be used to help guide us in those sorts of decisions. When you get to the end of your dose escalation part, we're also asked people to really take a close look at their clinical pharmacology, their PK data, their PD, any biomarker data they've generated, as well as the activity and safety data. And we recommend people do a pharmacometric exposure response analysis that incorporates all of this information so that you can make more rational selections about the doses that you're taking forward into your later development. Okay, so how do all the clinical pharmacology studies on the standard comment sheet enter into this? Well, in a sense, each of those studies is like the piece of the puzzle, and once you put it all together, then you can see the whole picture with respect to behavior of the drug. Now, another way of looking at this is in these terms. So if we're looking at the exposure response for the drug, we know that all of these different clinical pharmacology issues can have an impact either on the exposure or the response or both. And when one of these things changes, that can make you rethink how maybe you want to dose the patients or perhaps adjust doses in specific circumstances to address these issues. So, for instance, if you have a drug that's metabolized, you may have an in vivo drug-drug interaction liability. So that can lead to changes in the exposure of the drug. And with a consequent change in the response. So under those circumstances, you might need to adjust the dose for these patients. Okay. Similarly, renal impairment, hepatic impairment, these can all have an impact on this exposure response, as can many of these other issues shown over here. All of these issues that are listed here are the items that we have listed in those standard comments to prompt you to think about how these might interact with the exposure response for your drug and what you might need to do about adjusting the doses for those particular patients. So all of these CP issues then can really end up affecting the dosing in one of two fundamental ways. So if we go back to our exposure response curve here with efficacy and safety, some of these events can cause a reduction in the exposure of the drug, right? And that can lead to a coincidental decrease in the efficacy of the product. And that could be problematic for the patient population that we're looking at. Alternatively, some of the events I've described can lead to increases in exposure from the ideal. And although you may see some improvement in efficacy, oftentimes you're on the flat part of that curve and we don't really see very much. The greater concern is that there is an increase in the toxicity, and so the patients may experience more adverse events. And so regardless of what these issues are, we're trying to understand how they manifest and what we might need to do in terms of changing the dosing in these situations to offset the reduction in efficacy or increased toxicity. Okay, so which clinical pharmacology studies do you need? Do you need to do all of them? Well, the answer is probably not, and it really depends on the type of product that you're developing and the route of administration, and probably even the therapeutic area in which you're developing the product in. So, for instance, for a small molecule drug, we have quite a number of issues that we probably need to check on for their impact on the behavior of the drug in vivo. But a lot of these might not apply to a therapeutic protein, for instance, a biologic. So, for instance, these kinds of products are generally not metabolized, so you don't need to do those studies, and you're not going to have any drug-drug interaction liability. So these are not issues for you, and you wouldn't look at them for a biologic. On the flip side, there are things that apply to biologic that you don't see for small molecule drugs. Immunogenicity is one of those. So immunogenicity can impact the safety and the efficacy and the PK of the biologic, but it's never an issue for small molecule drugs. So you wouldn't look at that for those, right? Now, some of the issues you're going to need to look at based on the route of administration. So, for instance, for an oral drug, food can have a big effect, and acid-reducing agents can have a big effect. But if you take that same drug and you make an IV formulation out of it, then the impact of food goes away, as does CDER impact. So you wouldn't need to do those studies there or for biologics, for instance. Now, even for an oral drug, considering the things that it needs, you may have some drugs that have no metabolism, for instance, right? And if that's the case, then there wouldn't be any drug-drug interaction potential. You wouldn't need to do that study. And since there's no liver involvement in the metabolism, then there's probably not going to be any hepatic impairment issue either. So you wouldn't need to do those kinds of studies. So you really have to understand the basic characterization of your drug. You know, an example of this is Zometa. Zometa is a drug that's not metabolized, so there weren't any DDI studies. It was renally excreted, and that study needed to be done. But we didn't look at hepatic impairments or things like that. So which studies you do depends on the characteristics of your drug. Now, another consideration is the indication that you're developing the drug in. Okay. So even in the case where you do have some metabolism and you may have some hepatic impairment or the possibility of some hepatic impairment liability here, if the drug is being developed in a therapeutic area where patients with hepatic impairment are not eligible for treatment, then it wouldn't make any sense to look at those studies. Okay. So which studies you need would depend on really the type of molecule it is, the route of its administration, its basic characterization, and the therapeutic area that you're developing it in. So the next question that comes up is when do you do these studies? Okay, and so this is one possible scenario. I want to highlight there are different ways of doing these studies and this is just one possibility. When it comes down to it, you're going to have to make decisions that are specific for your product. Okay. So here we have drug development on the top here, and one of the things I want to point out is that many drugs in oncology are approved via the accelerated approval pathway early in phase 2, so I sort of equate phase 2 and phase 3 in this situation as being very similar to each other. Now, many of these studies are done even before you start human studies, right? So we often or usually have analytical methods that are validated before starting human studies. We often or usually do metabolism studies in vitro to understand whether there are any active metabolites that have to be assessed. And we do a lot of the P450 screening quite early to find out whether any of the P450 enzymes are involved in the metabolism of the drug, which might indicate the need for in vivo drug-drug interactions studies in development. Now, these were actually used to be done quite late in development. There are some reason why you could and maybe should look at some of these at least earlier in development. Mass balance studies were sort of a small study that was done that gave us an indication about whether the route of elimination was hepatic or renal, and suggested whether we might need to look in organ dysfunction for these patients. Food effect studies were done really from phase 1 all the way through phase 4, but I'm going to show you some information later that strongly suggests we really need to be looking at these early in development, early when we're looking at the dose selection in our patients. Now, as I mentioned, the hepatic, renal, and bioequivalence studies for new formulations that are developed in the course of the development process are usually done later in development. Okay. QT liability used to be assessed quite late. More recently, I think we're seeing this being done earlier in development. And importantly, pediatric patients. These studies are generally done after the drug has been approved in the adults. And most of the clinical pharmacology information in adults is bridged to patients either with a combination of an efficacy pharmacokinetic study or based on pharmacokinetic extrapolation. Okay. So this is one paradigm. It changes for each product. And as I said, you're going to have to make some decisions given the specifics of your particular product as to when and how to do these studies. I want to point out that your choices do have an impact on how well the development goes. And let me give you the example of Zycadia, which is approved for non-small cell lung cancer. And early in development, they selected a dose of 750 milligrams, which they wanted to administer on an empty stomach. And that's what they did through the development. But what we found out was, as this got later in development, there was a lot of GI toxicity associated with that approach, right? So a lot of diarrhea, nausea, vomiting, that sort of thing. And anecdotally, there was some information that if you administered this with food, you might alleviate some of that toxicity. Now, during development, we knew that food actually affected the exposure of Zycadia. It increased it by 75%. And so the desire was to avoid that during development so that we didn't have to deal with extra adverse events. But we saw the GI toxicity issue. And we wondered whether we could capitalize on this particular phenomenon by dosing Zycadia at a lower dose with food to achieve the same thing. And so we asked the company to do a post-marketing study on this. And that's exactly what they did. And this is the result here. This is the plasma concentration time curve. You can see Zycadia 450 milligrams with food shown in red versus Zycadia 750 milligrams fasted as indicated in the labeling, right? So when you looked at that, you could see that the exposure of both was basically superimposed upon each other. That was, in fact, pretty much what we expected to see based on what we knew about the pharmacokinetic behavior of the drug. Now, when you went and you looked at the adverse events, you could see that when Zycadia was given at 450 with food, it actually had less diarrhea, nausea, vomiting, and abdominal pain than Zycadia does when you give it at a higher dose under fasted conditions, right? So you can see that these are much higher here. And so we ended up changing the label, and currently we say take 450 milligrams of Zycadia with food. Okay. And so the question that arises is, would it have been better to do this dose exploration with food earlier in development? Well, it took us 3 years to get here, so personally I tend to think the answer to this question is yes. Okay. So when you're in early development, you're looking at dose selection. Some of the clinical pharmacology studies that I think you should be thinking about very early are the food effect question, right? So for an oral drug, from the very beginning, this is a consideration. Do you have a food effect or not? If you have a food effect, do you need to try and avoid those issues? Or can you improve your outcomes by giving your product with food? These are the sorts of things that you should be trying to address very early in development so that you can maximize this as you proceed with your development. Acid-reducing agents are another issue that you should think about early on. In many indications, patients are pre-medicated with PPIs or H2 antagonists because of the GI irritation. And we've seen in a number of cases that these agents can also affect the absorption of the drug of interest. So if you're going to be developing this product in a population of patients who are going to be treated with these kinds of drugs, you should probably look at these kind of studies early in development as well. And then there are drug-drug interaction studies, right? So there are a number of clinical situations where people are going to be treated with co-medications to treat some other comorbidities. AML is an example in which they're treated with antifungal agents. And antifungal agents are very strong inhibitors of CYP3A4 drugs. And so they can lead to excessive exposures of the drug. And if you had developed your drug or selected your dose in your phase 1 trial without taking that into consideration, when you moved into patients, you might end up with a situation where they are experiencing drug exposures and toxicities that are much higher than anticipated. And so this is something that should also be considered early in development. Now, what about rare diseases? Now, all the diseases in oncology are rare by definition, but we're talking about cases where the incidence is really low and there are really very few patients to enroll in studies. Do we still need to do all these clinical pharmacology studies? Well, the answer is we still want to optimize the dose in these patients, and we still want to leverage the clinical pharmacology because all of the issues that I've described so far also apply to the drugs that are developed for these indications. But it's a valid question, you know, how do we do these kinds of studies if we don't have the patients for them? Well, really, there are different approaches that we can use. Some of those are what we call model-informed approaches or model-informed drug development. And some of these are alternative ways of conducting these studies and perhaps doing these in alternative populations. And so I have a couple of examples to share with you. So ibrutinib was approved for a number of lymphomas some time ago. And early on in the original application, a drug-drug interaction study indicated that strong CYP3A4 inhibitors cause the drug exposure to increase 24 times. That was huge, right? But then we thought about moderate inhibitors, which had not been studied. And the question was, you know, even if they cause somewhere between a 5 or 10-fold increase in exposure, that's also very serious. And the question was, do we need to go and do an in vivo study to assess this? Well, actually what was done was some physiological-based pharmacokinetic modeling. Using the information from the strong inhibitors, the strong inducers, the company was able to develop a model that was able to describe the drug interactions that could be experienced. And so we were able to pose the question about what would happen with moderate inhibitors. And based on the results of this modeling, we actually provided dosing instructions in the label for those kinds of situations without the need for going to in vivo studies. And really, we are starting to see a lot of applications of PBPK emerge in areas like food effect studies, renal impairment, hepatic impairment, and also exposure response modeling and simulation is something that we are seeing a lot of. A very important tool is helping us to optimize doses as well as to come up with more efficient trial designs which save us time and money in the long run. So this is becoming a very useful modality for us. Okay, so we also have examples of using alternative approaches to get the information that we need to answer those questions. And the example I'm going to use here is pemigatinib, which is a drug that was renally excreted. And so the question was whether or not renal impairment had a significant impact on the exposure of the drug in the patients. And if you remember from a few slides before, I've depicted this as occurring in really a standalone dedicated study. And those can be difficult to do in some respects. But in this case, what they did was to broaden the eligibility criteria in their efficacy trial so that they could allow enrollment of patients with mild and moderate renal impairment. And what that allowed us to do was assess the impact of renal impairment on the PK in the efficacy trial. And we also had some additional safety information around that helped us to answer that question, at least to some extent, for patients with mild and moderate renal impairment. And that was a bit of a novel approach that we're starting to see more and more of in the applications that we get. Now, another alternative, as exemplified by venetoclax, is the use of different populations than the indicated patient population. So if you think about the original question, what do we do in a rare population that has few patients? Sometimes what we can do is we can do these studies in different populations and bridge that information to the indicated population. So with venetoclax, we were able to do some of the studies in healthy volunteers. The bioavailability studies, food effect, and the inducer studies were done in healthy volunteers, and we could bridge that information to the patient population. When you can't use volunteers, you can go and often use different cancer populations to do these clinical pharmacology studies, and usually findings in those particular settings can be extrapolated to the patient population of interest. So a question that we get often is whether we can do these studies of PMRs, and really the FDA cannot agree to that request because when you submit your application, it really needs to be complete in all respects. And in some ways that was a little problematic because that often meant renal impairment, hepatic impairment, drug-drug interaction studies. These were often pushed out and assessed post-approval. So we didn't have the information to provide the appropriate dosing instructions at the time of approval, which is a little problematic. However, by using these alternative approaches that I just described, different patient populations, incorporating these studies into other studies, we're at a point now where we're getting more information in the original applications that help us to provide appropriate dosing instruction for these patients when the drug is approved. Okay, and so as you go through, you'll be confronted with the same sorts of situations, and you'll have to make some decisions about that. I do want to ask you to remember that these may be small populations of patients, but they often are going to need dose adjustments to help them deal with toxicity or the possibility of a reduced effectiveness. So I'm going to leave you with a couple of takeaway messages. And the first of that is that really what we're trying to accomplish here is the selection of the best optimal dose for the patient population. The clinical pharmacology studies that I've described aim to do that, not only selection of the optimal dose, but also to come up with the dose adjustments for patients who have particular situations that need to be addressed. And finally, the third point I'd like you to remember after today is that there are different ways of approaching these clinical pharmacology issues and getting the answers to those questions. And so with that, I'm going to wind up my talk. And so I'll leave you with this particular closing thought, and that is that early interaction with the FDA generally will help in the development of your drug product. So I'm going to end here, and I will take questions that are submitted. Thank you very much.
Thanks, Brian. That's just the talk I needed at the end of the day. Very peaceful and calm. I'm going to go right to the audience questions. And the first question here, try to go through a lot of these. Is it possible to start first in human studies under the Fed condition? Can the same starting dose selected based on ICHS-9, standard dose 10, and highly non-severely toxic dose be used?
And Brian, hi, this is Jeff Kelly. It looks like we may be having some issue. We saw you come off mute, but we're not hearing anything just yet. Can you check to make sure that, there we go.
All right, there we go. Sorry about that. So thank you, Jeff, for the question. Very interesting. So I don't think we have seen anybody start off their first in human study right away with in the Fed condition, and I'm not sure that I would want to do that. I think when we begin, we're trying to get an assessment of what the systemic exposure of the drug might be. So we want to see what things are like in the Fed condition, in the fasted condition rather, and then perhaps later, as you escalate, you might incorporate a cohort of patients somewhere in your dose escalation where you do administer the drug under the Fed condition, so you can see what the exposure would be under both states. Another thing we have to worry about a little bit is that oftentimes food will increase the absorption of the drug, and you'd be worried about, if you gave it food and it had a big food effect, you'd suddenly see a very big change in the exposure, a big increase. And that could be problematic because it could cause more toxicities than you're willing to put up with. So if you did this really high or close to your MTD, that could lead to some problems. So probably doing a Fed cohort in the middle of the dose escalation is probably a good approach to take in doing that.
Great, thanks. Here's another question. And as a pediatric oncologist, everything was BSA. I was always surprised when people did kilograms. But the question is, can you please comment on flat dosing versus BSA or weight-based dosing? What are some considerations?
Sure, yeah, that's another good question. So really, can you give a flat dose versus a dose that's normalized for patient size? Well, this is a consideration you should be looking at regularly. So whenever you're developing your drug, you should be taking a look to see what the impact of size has on the exposure in a range of patients, okay? So typically when you move to a flat dose, often what you see is that patients who are heavier end up having a lower exposure than sort of your average size patients. And then at the other end of the scale, patients who are smaller tend to have higher exposures. And so the concerns there would be that in these smaller patients with these higher exposures, they may end up having higher rates of adverse events, and so that could be problematic. And at the other end of the scale, no pun intended, folks who are heavier might have a lower exposure, and you might suspect that could have an impact on efficacy. So you really, for whatever, anytime you're developing a product, you should be assessing this to see how that shakes out. Okay, if it's excessively high exposures in small patients, that could be problematic. You might need to cap the dose in those people, and same for the folks who are heavier.
Got it. Thanks. But I assume flat dosing is much later down the road when you want to make it convenient for the patient. But initially, if you're really looking for responses, you want to optimally dose, and at least optimally dosing would be based on BSA or weight, right?
So yeah, that's a very common practice. Is people will start out with a weight or size-based dosing, and they'll often want to convert to a flat dosing paradigm as they get later in development.
Thanks. Another question here. Is it expected to have food effect data in advance of a phase 2 study that is anticipated to be a pivotal study? You know, in today's world, phase 2 is phase 4, phase 3.
So yeah, that's a very good question as well. So, you know, if you have an oral drug, food is always going to be a consideration for you. And really it's best to make these assessments early in development because you got to make some decisions about how you're going to dose your patients. You know, should they be taking this with food or not? That's going to be important information that will get translated not only into those efficacy trials, but also into the labeling for the product, you know, so we can inform patients of how to take this effectively and safely. So if you have an oral drug and you think you're going to be doing a lot of your pivotal studies in phase 2, then you probably want to be taking an early look at the effect of food in your phase 1 studies to make some rational choices about how to dose those patients moving forward.
So there's a specific question about one of the slides for the ADME mass balance study is done very early. What is the consideration for that, and in what scenarios we can prioritize or deprioritize the study for small molecule oral drugs?
Another good question. So oftentimes mass balance studies are done very early on, and they give us 2 bits of information. One is they tell us about the metabolites that we may need to be looking at in the patient population. They also give us information about the routes of excretion for the drug. Now, we've had some cases in the past where folks have foregone doing the mass balance studies altogether based on the approach that they're going to measure the parent drug and its metabolites regardless. And they committed to doing renal or hepatic studies from the outset. And that's a legitimate approach. Another thing you might think about, though, is that the mass balance study often provides information that's very useful in helping to build your PBPK models. In fact, in the case of Loxator, that was the drug that was approved for sickle cell disease. Based on the mass balance study, they were able to refine their PBPK model, and using that information in conjunction with the in vitro data, we were able to use PBPK modeling in that particular case to forego in vivo drug-drug interaction studies. So the model was so good, and we knew so much about the elimination pathway that we were confident in that modeling such that we didn't need to do any drug-drug interaction studies in patients, and we were able to provide appropriate dosing instructions in the labeling for that product. And that was an exception, I have to say. But yeah, that's another consideration you might want to think about as you move forward with your product.
Thanks. Back to another question on one of the slides. It was interesting that there was a drug that went through trials, wasn't approved, and then it got approved at a higher dose. So clearly, how did they miss the right dose, the effective dose? What was the general gestalt that happened there?
Oh, geez, that goes back quite a ways, and I'm not sure I quite remember all the details about it, but in their earlier trials, they had found that they had the 2 milligram per meter dose was what they found effective in the earlier trials. But then when they moved into their efficacy trial, it didn't pan out so well. And unfortunately, that company then gave up on the product, and another company bought it. They then redeveloped it, and knowing that the 2 milligram per meter dose wasn't effective, did studies at a higher dose, and it worked out for them.
Okay, thanks. The healthy volunteer studies with the venetoclax. Most of our oncology drugs, and I have to go back and look up venetoclax again, but I mean they're relatively toxic, and we tend to keep them out of healthy volunteers. Did we use a subtherapeutic dose that was expected not to be toxic, but would give us some kind of PK data for some kind of organ impairment to use that data? Was that the general approach there?
So when it comes to doing healthy volunteer studies, clearly the toxicity of the drug is a big consideration, and a lot of people weigh in on the suitability of using that approach. The other considerations are whether these are genotoxic or reprotoxic or things like that. And as you said, in many examples that we have, they aren't suitable for doing healthy volunteer studies. In the cases where they are, oftentimes we can use the doses that are indicated or doses that are near what are indicated. One of the considerations would be that if you need to use a lower dose for safety reasons, we would be looking at something that would be in the pharmacokinetic linear range of the drug. And that's how we would approach that.
Got it. I know I've seen this. I know that this issue has come up. Instead of, I want to get my drug approved. I don't want to do renal or hepatic impairment studies. Can I just contraindicate those in the label and be done with it?
That's another good question. We've seen that one in the past once in a while. And although it looks like an attractive avenue to pursue, we actually can't take it. And that's because the Code of Federal Regulations prohibits or limits the contraindication that you can put in the label. That contraindication has to be based upon an observed adverse event, and that adverse event has to outweigh any therapeutic benefit that you might get from the drug. So it can't be a theoretical possibility. So if you had a drug that was renally excreted, you couldn't argue, let's just contraindicate renal impairment, because we have cases where renal impairment maybe increased systemic exposure a little bit, but it doesn't lead to any clinical issues, so you wouldn't need to do anything. And you don't know that until you do those studies. Okay. Secondly, we probably really don't want to take that approach anyway, because we don't want to limit any therapeutic options for patients. So if you had that drug with renal elimination, we would want to explore the impact of renal impairment so we could better understand if the exposure does change significantly, whether we need to come up with a dose that's more suitable for those kinds of patients.
Yeah, I should have pulled out the contraindication right away and used something like a limited use indication or something, so that we wouldn't have had that word contraindicated in there. But no, I completely agree with those points. So that is the end of the time for discussion and questions and answers here. I want to greatly appreciate you taking the time to join us and giving us that presentation. And I'm going to turn this back over to Lisa, who I think is going to welcome Mark to